Hi, everyone. Good morning. We're going to go ahead and get started. That way we can have ample amount of time for questions at the end. Thank you guys so much for joining us for Grand Rounds this morning. We have a fantastic lecture by Dr. Spec. Just some background information on Dr. Spec. He went to medical school at the University of Illinois College of Medicine. He then went to residency at Rush University Medical Center before joining us at WashU for fellowship in infectious diseases, where he also obtained his master's in science and clinical investigation. He is now an associate professor of medicine in the division of infectious disease and has quickly become our fungal expert. So let's welcome Dr. Spec.
Thank you all for the invitation today. Thank you, Chiefs. Disclosures are up here. As I was pointing out, they're not the fun ones. Essentially, the disclosures that I have are studies that we're doing, as well as me helping companies design studies for drug development. And the only drug I'll be talking about today is not FDA approved, so there will be nothing you can prescribe today. So we're going to talk about nothing left but the decomposition, stories from the fungal apocalypse. So this is essentially, I want to thank Abby Sung for coming up with the idea of this lecture. As the craze of Last of Us swept America, she said, can we have a lecture by you to tell us, can a fungal apocalypse actually happen? And if it does happen, what would it look like? So for those of you who have not watched The Last of Us, I'm going to leave you with the opening scene. That's what we're going to start with.
Mankind has been at war with the virus from the start. Sometimes millions of people die, as in an actual war, but in the end, we always win. There are some fungi who seek not to kill, but to control. Viruses can make us ill, but fungi can alter our very minds. There's a fungus that infects insects, gets inside an ant, for example, travels through its circulatory system to the ant's brain, and then floods it with hallucinogens, thus bending the ant's mind to its will. The fungus starts to direct the ant's behavior, telling it where to go, what to do, like a puppeteer with a marionette. And it gets worse. The fungus needs food to live, so it begins to devour its host from within, replacing the ant's flesh with its own. But it doesn't let its victim die, no. It keeps its puppet alive by preventing decomposition. How? Where do we get penicillin from?
Fungus.
Oh.
Dr. Schoenheiss, you're in distress.
Fungal infection of this kind is real, but not in humans.
True, fungi cannot survive if its host's internal temperature is over 94 degrees. And currently, there are no reasons for fungi to evolve to be able to withstand higher temperatures. But what if that were to change? What if, for instance, the world were to get slightly warmer? Well, now, there is reason to evolve. One gene mutates, and an Ascomycetia, Candida, ergot, Cordyceps, Aspergillus, any one of them could become capable of burrowing into our brains and taking control, not of millions of us, but billions of us. Billions of puppets with poisoned minds, permanently fixed on one unifying goal: to spread the infection to every last human alive by any means necessary. And there are no treatments for this, no preventatives, no cures. They don't exist. It's not even possible to make them.
So if that happens,
we lose.
we'll be back.
I'm going to be slightly less dramatic, but they will tell you a few things that are wrong with that. Obviously, we do have fungal infections in humans. Most of you have had at least one case of them in your life, or at least one case you cared for. And many of you living in St. Louis have actually had a fungal infection, because many of us actually carry histo in us right now. But what I found the most amazing, other than the spot-on costumes, was the fact that both the physicians were smoking on TV. So let's move on to...
Mankind has been...
Oh, we're going to watch that again. So this actually, the popularity of The Last of Us as the first episode premiered was so popular that essentially every news agency in the world covered could a fungal apocalypse happen? And I think I received about 300 interview requests, and I fled quickly. But essentially, this has catapulted into the mind, into the zeitgeist of society, what could happen with a fungal apocalypse. And the one thing that I want to, for those of you who have watched The Last of Us, I want to contrast it to the video game, because The Last of Us is actually based off of a video game, which is actually much more accurate in a very important way. This is a screenshot from the video game. Funguses don't spread like foodborne illness. They spread through spores. These spores can reach the stratosphere. So it is not, so Aspergillus that starts in Kentucky can infect somebody in Thailand. Not all of them do this, but many do. That is a fundamental difference. But the reason they chose to do it differently in the show is because they wanted to focus on the good acting, on the emotive capacities of the actors, which are impressive. It's really hard to do that with a gas mask. Like, it's really hard to have an amazing scene where you express the dread that you're feeling while you have a gas mask. But essentially, that would be the issue. Fungi form spores. These spores are everywhere. And one of the reasons why we gave up on using bubble boy rooms, the clean rooms for bone marrow transplant patients, is because they don't work. Even HEPA-filtered rooms have fungus in them. We, on average, if you do the math of different environments and how many mold spores they have, and then you multiply that by the tidal volume of an average person, multiply that by the number of breaths in a minute, minutes in a day. On average, we inhale, depending on where we are throughout the day, between 50,000 and 500,000 mold spores a day. So the fact that we are not constantly infected with fungi is really just a marker of our immune system and about the body temperature at which we live, as the guy in the TV show pointed out really well. So let's talk about the fungus that he talked about, so we can get that out of the way. What is cordyceps? So cordyceps is an ascomycete. It's actually a family of ascomycetes. There are thousands of them, actually tens of thousands of them. They are very host specific. They infect the host, and what they do is, as he described, they literally infect the brain. They flood the brain with hallucinogens, and they force the insect, or it's actually mostly, it's arthropods, it's more than just insects, to spend more and more time at higher and higher elevations. And so you'll see ground-dwelling insects like this cricket that'll climb a tree. And eventually, as they get older and older and older, the fungus will start to grow out of its feet and root it in place. And they will stay there until they die and are fully consumed by the fungus, which will then fruit. These are the ascomycete fruiting bodies that you see here. These are essentially evolutionarily analogous to morels, which are delicious. And they will spread these spores. And the reason they want to be high up is they want the spores to go farther and farther and farther, because these are host specific, so they need to be able to reach the next host. And this is mostly seen in the tropics. So the question is this something that you're going to see in St. Louis? Or is this something that we see, like, you know, South America, Africa? This is a picture of a daddy longlegs. So daddy longlegs, if you ever go in your basement, especially if you have a crawl space, if you have a dank, dark place, you will often find what they call them mummified daddy longlegs. That's cordyceps. There's a cordyceps that infects daddy longlegs in Missouri and all of the United States, actually. And you will literally see them. Often what they will do, you chose this picture not because it's representative, but because it's beautiful. It's a National Geographic picture, actually. But what they will usually do is they will spin a web, they will hang off the web, and they will fruit in that position. so that they can swing in the breeze and spread the spores. But is that the only thing that cordyceps does? The answer is no. Cordyceps is actually can also decompose plant matter. Some species do that. This is actually a cordyceps that is commonly grown commercially. So this is a recipe. If you happen to want to grow your own home cordyceps, your zombie fungus at home, you can follow this recipe of a little bit of egg whites, some malt extract, yeast, some water, and some rice grains, and you can brown rice. Definitely, you want the organic brown rice. The fungus only eats the best. And you can grow your own cordyceps, and you can be like, hey, Mom, look, I brought you a zombie fungus. We even use them as food. This is actually a picture from the menu of a Michelin star restaurant in New York. In that pasta, the orange pieces are cordyceps. We eat cordyceps. It has been used for millennia in Chinese traditional medicine. My favorite experience with it was one of our nurse practitioners bought a coffee substitute, which I don't know why, but she bought a coffee substitute that made of cordyceps, which she absolutely hated. And then went with, well, you like fungus, here you go, and gave it to me, and I tried it, and it was truly horrendous. Just in case you're ever wondering about getting a cordyceps-based coffee substitute. So this is how they live in the environment. So could they do what they did in the show, where they jump from one of them jumps from an arthropod into a human and then causes a fungal apocalypse that wipes out everybody except for like 6 people? You can't hire too many actors. The answer is no. We are actually we are deuterostomes. Insects are actually protostomes, which means that we are so far removed from each other. It's about 600 million years of evolution. The only animals that are farther away from us than insects and other arthropods are Cnidaria, so corals, medusa, anemones, and then sponges are even further removed. But if you really think about that, this is an organism that has evolved over 600 million years to infect a particular host, which is then going to jump 600 million years of evolution to the next host. No. Like the number of genes that are involved in that harmonious dance of pathogenicity are just way too wide. So this will never happen. We will never have a cordyceps zombie fungus that takes over humans. All right, we're done. I'll talk to you later. I'm kidding. So let's talk about fungus and why a fungal apocalypse is actually something that should be considered. So the first thing I'm going to talk about is in situ evolution of fungi. Fungi are masters of biochemistry. They are the producers of most of our antibiotics. They're the producers of most of our antifungals. They're the producers of most of our earlier chemotherapies. Now the target-driven therapies are very different. But if you look at what group of creatures has given us the most drugs over time, it's fungi. They're the masters of biochemistry. Their biochemistry is some of the most complex in the world. This is one of my favorite phenomenon. This is called Titan cells, or in this slide, they call it giant cells. But Titan cells is the name that the literature has settled on. What happened, most fungi, when they encounter a hostile environment, and if you're being attacked by the immune system, if you're being given azoles, those are hostile environments, will actually have a whole suite of genes that they will upregulate to change their evolution. They will actually increase their mutation rate, not as a byproduct, but as an intentional product. This is a Titan cell, and what that means is that it's multiple copies of the fungal genome. So if you've ever eaten a strawberry, now they're the size of an apple, right? But if you ever go picking wild strawberries, they're about this big, right? They're tiny little things, and it's the same amount of flavor, just a much smaller package, right? Well, those smaller strawberries are 2N. They have 2 copies of the chromosome. The current strawberries that we eat in the market are between 64 and 128 copies of the chromosomes. Because with a plant, the more polyploidy events you have, the bigger they get. Same thing happens with fungi. If you create polyploidy events, the actual cells get bigger. But what's really amazing is that when you have a strawberry, it's 128N. That is never, again, going back to a 2N strawberry. It just, that if you think about the complex mitochondrial spindle, back to like biology from high school and college, that is not happening, right? Like that's just not gonna happen. How do you create that? Don't know, but Cryptococcus does. So what happens when Cryptococcus enters a human lung is that it forms Titan cells, which are over 1,000 copies of the chromosomes. And then it spits out daughter cells that are haploid or diploid, that have tons of chromosome breaks. And the most common thing that happens is you have chromosome breaks from regions of chromosome 1 and chromosome 3. Chromosome 1 and chromosome 3 contain the polysaccharide capsule. They contain the chitin genes and a few other pathogenicity islands. So basically what happens is you get a fungus that enters the body that's adapted to live on a tree or in a pill bug, which is designed to evade amoeba. And by the time it's done with a month in your lungs, it's now designed to pathogenize humans, including creating these large cells that you'll see here, which are very large capsules. So is there anything else that's helpful for being so big? They're bigger than macrophages. They physically cannot be swallowed. Because they also have so many copies of genes, they're also highly drug resistant because many of those are mutated. So this is one of the mechanisms by which Cryptococcus will persist forever in some people after they've been treated. Doesn't mean they necessarily cause pathogenicity, cause infection, but nonetheless, they will persist forever in some people. Because the other thing that's important to realize is that why do pathogens pathogenize, right? A virus pathogenizes because it needs to. It can't reproduce without it. Bacteria are evolved to take advantage of what they can. So they end up in the wrong place, the wrong time, they make do. Why do fungi pathogenize? When Aspergillus gets into our lungs, when Histo gets into our lungs, Cryptococcus gets into our lungs, they don't leave our lungs and then infect another person. The only cases of fungal human-to-human transmission of things like that are actually in solid organ transplants, when we actually transplant the lung. And evolutionarily, that's not a very good strategy. So why are they there? What I like to tell people, never say this to a patient, is that essentially a fungal infection is pre-decomposition. They want to decompose us. They want to be there so that when we die, they can start decomposing. That's what a fungal infection really is. It's pre-decomposition. And if you think about it, once you run out of your immune system, you're just really kind of a warm body to pre-decompose. And that's what fungi are trying to do. This is a wonderful work by Malcolm Richardson from across the pond in Manchester. This is actually sequencing of Aspergillus fumigatus, true fumigatus, we'll talk about that a little bit later, from a single patient who had chronic pulmonary aspergillosis for 7 years. So this is 7 years of infecting a human being with exposure to drugs, etc. The red box is all of the known variants in the world environmentally of Fumigatus sensu stricto locate to the red box. All the rest is brand new isolates that have more diversity in one single patient than all of the environmental isolates that have been found in sequence to date. That's the kind of evolution we're talking about. So this rapid evolutionary adaptation has been found so far. I'm not going to go through all the examples. In Candida, Aspergillus, Cryptococcus, Coccidioides, and Mucor. We have not yet looked into a fungus where we failed to find it. So maybe it doesn't exist in some, but we have not found it in any. So have there ever actually been fungal apocalypses, right? We're not going to talk about the Salem witch trials, which was a ergot intoxication, but let's talk about real fungal apocalypses. The big one, Cotricomycosis. It's been called multiple times the amphibian apocalypse. And this is actually aptly named. But basically what's happening is there's increased evaporation that leads to increased cloud cover across the world, which decreases daytime water temperatures in the tropics. It's not a huge difference, but it's enough that the body temperature of frogs every year at the peak, so the peak body temperature is lower than it used to be, by a couple of degrees. Nothing huge, right? Except that now this fungus, which they can normally could clear, they can't clear anymore. It's led to a dramatic decline of 200 species across the world. 90 of them are now extinct. We have, in the last 20 years, watched 90 species of amphibians go extinct because of one fungus. That's the definition of an apocalypse, right? We have had absolute extinctions of these species. And the most amazing thing, and we're going to go back to this multiple times, we can cure these frogs. Itraconazole actually works really well, but we're not going to environmentally give itraconazole, right, to the frogs. But if you take these frogs from the environment that are, infected to the point of they're not going to recover, because once they infect and show any signs, the mortality is 100%. You put them in a lab conditions, and over about 2 days, you increase the temperature to about 38, 39 degrees Celsius. Fungus dies. Fungus is not heat tolerant. That is the biggest hurdle that keeps fungi from being able to decompose whatever they want. Most fungi do not survive at 37 degrees Celsius. They can't really, as the person spoke really wonderfully in that clip, they really struggle above 34. About 34 Celsius is kind of a peak for most fungi. Then we can talk about the bat apocalypse. You may have heard about the white nose syndrome. And this is a quote from the USDA. The white nose syndrome is considered one of the worst wildlife diseases in modern times, having killed millions of bats across North America. This is one of the bats most commonly affected is called the little brown bat. Great name, little brown bat, very imaginative. However, their population has dropped 97%. And many people in this room are probably going to go, who cares? It's a bat. How about if I told you that one bat can eat up to 500 mosquitoes every night? That's their food. They eat mosquitoes. And the actual rates in those places that have been infected by white nose bat syndrome have had dramatic increases in mosquito populations. So when you're getting consumed, and St. Louis is one of those places, so when you're getting consumed in the summer, you can thank a fungus because the bats that eat them are gone. 3 species have more than a 90% drop in their population in less than a decade. So if that's not an apocalypse, I'm not sure what it is, right? We talk about decimation, the most horrible thing you can imagine, decimate somebody. That's losing 10%. They're keeping 10%. And what really happens is that they die during hibernation. When they're not hibernating, the bats do fine. They can fight it off because their body temperature is high. When they hibernate, their body temperature gets lower. And what happens is as they're roosting here, as you can see, this is actually from the Sierra Nevadas, they get these fungus that come out of their nose and release all these spores. And they get weaker as they lose weight, as the fungus consumes more and more of their resources. The immune system fights it more and more and more. They get weaker and thinner, and their muscles get thinner, just like our patients who are infected with fungal infections. They waste away until they're so weak that they fall off the perch, and they're no longer able to climb back up. And they die. Funny thing, if you take that bat, bring it into the lab, raise the temperature to 37, let it break hibernation, feed it, it clears the fungus. It's a temperature. This is Arturo Casadeval. You may see that he kind of vaguely resembles the gentleman in the first story, in that first clip. He's a professor. He's the division chief of molecular microbiology at Hopkins. And he has been doing this work for a long time, amongst other things that he does, on fungi, heat tolerance, and climate change. And one of the things that he has proposed, and this one's far-fetched, might be real, might not be real, we'll never know, because really there's no good way, we don't have dinosaurs around anymore, is that as the asteroid impacted the Yucatan Peninsula, it dropped the global temperature. But because dinosaurs are endotherms, or many of them were at least, exotherms, they behaved a lot more like the frogs do in the previous extinction. And the fungi that rose up could very easily infect them and kill them. And so he's proposing that another hypothesis as to what killed off the dinosaurs is actually a fungal apocalypse. He has some data for it from sequencing of dinosaur skeletons. It's hard to say, like, is this pre-mortem or post-mortem decomposition, if we use that analogy that I used before. I don't know. I think it's interesting, is all I would say. But what's really scary is that what's happening now is that molds are becoming more thermotolerant. And it's not the fact that we have a little bit of a rise in temperature of 1 degree. That's not what makes them more tolerant. It's the heat waves. It's when northern France gets to 105 and stays there for a week. That's a bottleneck event. And the fungi that survive are the ones who are more thermoresistant, that have more heat shock proteins. So let's talk about the looming threats in fungi. So this is actually an alert that was put out by the CDC that largely went unnoticed, which is saying exactly what I was saying. If you look down here, is that as the globe warms, it is quite likely that fungi are going to become more thermoresistant. And there is quite like the concern that we in the mycology world have. And we think that this is actually how things like Candida auris came about to be, is that Candida auris always existed. It was always very hardy. It was always very resistant to drugs because it lives in a milieu where it was exposed to other things. And it's one of those hypermutable pathogenicity island fungi, which is actually, Candida auris is exceedingly hypermutable. What happens with that is that it gets warmer, it picks up that one thing it's missing, the one thing it needs to start killing humans. And now it's here. Now it's killing humans. This is a wonderful study from about 15 years ago from MBio that looks at a cost fitness curve. If any of you are ecologists, you can maybe explain it to me. I don't fully understand it because I haven't done advanced mathematical ecology. But essentially what it boils down to is if you look at the cost of fitness of being warm-blooded, it's high, right? We need to eat almost like pound per pound. A mammalian carnivore needs to eat almost 10 times the amount of food that a crocodile does, for example. So that's a big deal. That's a lot more food you have to find so you can reproduce. There has to be a reason for that. And if you map it out, how much you're protected from fungi versus how much you lose by having to do all that extra eating, the peak of perfection is around 36 to 39 degrees. It is possible that fungi are the reason we are warm-blooded. This is a case that we published a few years ago. It was a gentleman who had a fungi called Thyronectria austroamericana. Never heard of it before. Grew out of his knee. We sequenced it. Sure was that. The wonderful thing about Thyronectria austroamericana, it's a plant pathogen. It infects plants. It causes cankers on honey locust trees, which you can see here in panel D is what a honey locust is, and it's a honey locust canker. He actually had a honey locust in his backyard, and he was an avid gardener. And if those of you who may have had honey locust trees, they have thorns about that big. He would get scratched by them all the time. So he had a really bad septic arthritis from it. Gentleman in his 80s, no past medical history, no immunosuppression, none whatsoever. And then when I published this case, I got a phone call from a ER physician in Kentucky who was just diagnosed with tenosynovitis with Thyronectria austroamericana right after he cut down a honey locust tree in his front yard. This has never caused human disease before, and we now have 2 cases, all of a sudden. This is a CT from another patient that I've cared for. This one's a rural guy in Missouri. He grew a fungus called Glomus macrocarpum. Never heard of it before. Immunocompetent guy, mid-50s, huge brain abscess, as you can see. Had to go look up the fungus. It is a mycorrhizal fungus of plants. It lives in plant roots and fixes nitrogen. We sequenced it. That's the one. We did susceptibility testing. Highly drug resistant. Has it ever caused human disease before? No. So when we got the sequencing back, he was in my clinic and I asked him, what do you do? He goes, I'm a mechanic. Well, that didn't make a lot of sense. Like if you repair cars, you shouldn't get a agricultural fungus. Turns out he doesn't repair cars, he repairs farm equipment. He repairs tillers, tractors, combines, etc. This is a fungus that has never caused human disease before, that doesn't even pathogenize plants. It lives in the soil, but it had everything it needed to cause a brain abscess in an immunocompetent man, and it was highly drug resistant. Let's talk about everybody's favorite Candida auris. 2009, first case ever discovered. It's now everywhere in the world. And what's really amazing, unfortunately I didn't put this up, but I should have, it's not a single clonal event. There have been at least 6 separate clades that are separated by evolution by at least 10,000 years, which means that 6 separate clades minimum invaded humanity in a span of 5 years. This is the map of the United States. Missouri is colored in, but up until recently, we did not have any lateral spread in Missouri. Within the last 2 weeks, we have 2 cases. So it is here. It is now spreading in Missouri somewhere. We don't fully know where, but it's somewhere. So yay, it's around here and it's around stay. What do we know about Candida auris? 90% fluke resistant, and with the hypermutability, it needs a single point mutation to become fluke resistant. If it's not, I would never use fluke. You have a very high risk of failure. Elevated MICs to voriconazole. About 15 to 30% are resistant to amphotericin. Echinocandida, about 2 to 8% resistant. Multi-drug resistance, meaning 2 classes or more in 50% of cases. And all class resistant, 4%. The 4% resistant ones are spreading in the United States. There was an MMWR last year. We actually do have confirmed lateral transmission of multi-drug, or I should say, not even Cody drug resistant Candida auris. So literally no drug. And mortality at Barnes, I would say we're a pretty good hospital. At 90 days for having candidemia is 43%. With no drugs, it's 95%. And it's really hard to get rid of the environment. It hides, it plays peekaboo, it's highly resistant from many, many, many disinfectants. And within 4 hours of decontaminating the room, if the patient's still in there, about 1/5 to 1/4 of the surfaces are contaminated again. So very hard to keep out of environments. And the cases in the US are rising. This is going up to 2018. In 2020, a little thing happened around the world that kind of took the eye off the ball for most infectious disease doctors. A little thing, you may have heard of it, it's called COVID. And we really stopped paying attention to these rates. They're way higher now. That's just kind of, we had to shift. The other thing to worry about is azole-resistant aspergillus. It was reported only a few years ago in about a decade and a half ago in Europe. And now we kind of see it everywhere in the world. Some places have a lot of it, some places have less of it. And it has two mechanisms. One is what you call Aspergillus fumigatus is not Aspergillus fumigatus. You know, when you culture a bacteria, the way we identify it, if you have never really learned about MALDI-TOF, is we literally shoot it with a laser. And this is not an Austin Powers reference. Like, we literally shoot it with a laser, let the vaporized particles go through a matrix gel, and we identify the fingerprint of it. Like, this is science fiction, right? How do we identify a fungus? How do we identify a mold? Well, we let it grow. We let it produce fruiting bodies, i.e. spores. And then we take, and I kid you not, folks, Scotch tape, and we put Scotch tape right on top of those fruiting bodies, and we put that on a slide with some stain, and we go, it kind of looks like fumigatus. That's literally how we identify it. It's medical botany. You know, we're identifying it based on flower structure. And once you start sequencing, it turns out fumigatus is not a species. Fumigatus is a species complex with almost 100 species. Some of them are highly drug resistant. So when we find out that they're fumigatus and we give them voriconazole, they might be voriconazole resistant. We're also creating more resistance, both by encouraging those species and encouraging Aspergillus fumigatus sensu stricto to mutate. The reason I have a picture of a tulip on here is because this is where it started. It started in Northern Europe, and it was the Irish scientists who figured it out. We use azoles in crops. And the reason that we haven't seen a ton of Aspergillus resistance over the years is because we embalm people. So when a person goes through all those mutations and becomes highly drug resistant, they get embalmed, they get buried 6 feet under, or they get cremated, and that Aspergillus never makes it back out into circulation. But if you dump literal tons of azoles into the environment, you end up with drug-resistant aspergillosis. And that's what we have now. And it's in the circulation, it spreads, it's everywhere. And the one thing that's different about a fungal apocalypse that's worth noting, if it were to happen, is that a fungal apocalypse will not behave like a viral apocalypse, because a virus, as the density of humans goes down through the action of the virus, right? As you go from 7 billion people in the world, let's pick 8 billion people that we're now. Let's pick the most horrible virus you can imagine. Once you get down to 1 billion people, its spread slows down dramatically. But if the spread is from environmental spores, it's irrelevant how many of us there are. The infectiousness is just as high for the last human on the planet as it is for the first one. And that's actually why fungi have been so successful over eons at driving species into extinction. So will the new drug save us? This is one of my new favorite drugs, Olorifim, because it's done some amazing things for my patients. This is the drug I'm going to be talking about. It's not FDA approved yet, but I've had some amazing successes with it. So it's an active against dihydroorotate dehydrogenase. Inhibits DNA synthesis, so the way I like to tell people is the Bactrim for fungus, right? So it inhibits DNA synthesis. The binding against the mammalian analog is about 20,000 times lower, so there's no on-target effect. It's orally bioavailable, wide tissue distribution, orphan drug status for invasive Aspergillus lamentospore. I've seen this drug get patients who were dying for 6 months and have failed every single drug on the market, turned around in 4 days. Like, just dramatic responses. Very good activity against wide things. Doesn't have activity against yeast and mucor. And an important concept I need to tell you right now before we watch this little video here is the difference in molds between MIC and MEC. MIC is minimum inhibitory concentration, which is what we talk about with bacteria. Meaning, you give this much drug, the bacteria cannot grow, the bacteria dies. That doesn't happen really with molds in vitro. They're so fit to live in vitro that most of our drugs don't actually kill them. All they do is kind of slow them down and make them look funky. So the structure changes. So it's called MEC, or minimum effective concentration. So we're going to watch this video. of Aspergillus fumigatus incubated with olorofim. Starts to look funky after about 12 hours. You know, it's getting bulbous, getting a little funkier and a little funkier, and about 18 hours it pops. And then another one is about to pop. And if you watch this video for another 6 hours, you will see that all the cells pop. It is the most potent antifungal against Aspergillus that has ever been developed. And it works wonderfully about things like cocci, where it actually may have a chance of curing cocci-meningitis. We actually may have a drug that cures cocci-meningitis. We haven't been able to prove that yet because nobody's been brave enough to take these patients off of the drug. But essentially what happens is when they're given this drug, you have people who are moribund and about to die who just kind of wake up. Almost hard to reduce serial passage resistance, but there are isolates that are resistant naturally. And it looks very exciting. So I want to talk about, I'll skip this part and talk about the case. This is a case that we took care of. It's a lung transplant patient who was transferred to us from Oklahoma, I believe this gentleman was. 60 years old, lung transplant recipient. He has scopulary opsis brummtii. Wonderful name, pan-resistant mold. Absolutely every drug we have on it doesn't do squat. He has an anastomotic infection, airway obstruction. He had 5 weeks of combined therapy of Caspo, posaconazole, and terbinafine. And every 2 to 6 weeks, the lung transplant docs would have to go in there and laser through because he couldn't breathe. So the drugs weren't working at all. They literally had to mechanically debulk him once every month so that he wouldn't choke off. So we put him on olorofim. This is day -4. Day 10 and day 26. Gone. Remarkable response. It's currently up in front of the FDA to be approved with only phase 2 data as breakthrough therapy. The FDA has asked for a little more analysis of said data, but it's looking hopeful that we may actually have it approved by next year. So we might have a 2024 approval. We've already approved a pesticide in the same class in 2021. And you know where we're using tons of it? Almond orchards, which are in Bakersfield, California, where cocci is. So our most prominent, our most promising cocci drug is currently being dumped by the ton into cocci land. We're either going to exterminate cocci by causing an environmental genocide against cocci, or it's going to mutate. I'm betting it's going to mutate. And we're going to have olorofim-resistant cocci within a decade, wasting our most promising drug. On that cheery note, fungal apocalypses are not rare. They've been rare in humans, but they're not rare. It would look nothing like the show The Last of Us. The video game is more accurate. The fungi are extremely dangerous because of their unique biology. And if you look at the kind of apocalypse that we would be looking at from a fungus, it's exceedingly unlikely that we're going to get one. But if we do, this could be a true existential threat to humans. But we are literally currently experiencing multiple outbreaks of both novel diseases and drug-resistant versions of the diseases we have. So with that, I'm going to give you the slide that many of you care about the most.
Curious, there's been a lot of discussion in the cardiology there about therapeutic cooling after cardiac arrest. Investigation in the past that talked about using therapeutic heating for
So we do that. One of the treatments we use, for example, for sporotrichosis is heat pads. And they're mild, they're pretty effective. They are just a giant pain. The problem is that it's really that transition from environmental to pathogen where that temperature matters the most. Because for most species, by the time they're pathogenizing humans, they're thermal tolerant. It's that transition from the environment that temperature matters the most. Because if you take there was a wonderful study that was done by Nate Wiederhold out of Texas, where he took isolates that they had in their bank from environmental molds going back to 1970s. And they literally just took every single isolate that they had, put it on a plate, and incubated it at 35 degrees Celsius. And the idea was to see how many of them can survive. And the rate of survival was about 12% in the 1970s. And it's almost a linear rise since then. And now the modern ones, or relatively modern ones, I think this in 2015, they have about a 22% survival rate. So there's been a 10% absolute rise in thermal tolerance over a course of 50 years. It's a very short amount of time for that percentage of environmental molds to have picked up that resistance. But it's interesting if that would work. I think it's not as helpful as we would hope. Yes. Mm-hmm. There are many barriers to developing new drugs. The big one is that, we think of fungi. Fungi are often like, to this day, classification of fungi, the actual nomenclature, is in the same body as plants and algae. And that's how we thought of them historically. We thought of them as like plant-like. Evolutionarily, they are closer to us, way closer to us than they are to plants. And so that's a big issue because it is very hard. It's kind of like cancer. It's very hard to find a cancer agent that doesn't kill human cells. Well, it's really hard to build an antifungal that's not also a good anti-human because we have so many of the same conserved mechanisms. Azoles work, again, the reason they have drug-drug interactions is because we use their CYP3A4 mechanism, which is the CYP51 gene, which is the analog of the 3A4s, in order to, CYP450s, in order to kill it, because they use it to build their cells. So that's one issue. The other issue is cost. Fungi do not make the kind of money that antibiotics do. They certainly don't make anything resembling things like cancer drugs. And so if you talk there's tons of promising compounds out there. There's probably about 50 that I'm aware of. And you talk to venture capitals and they say, you never want to invest in antifungals. You double your money at best. Because when they're looking to invest in small companies, they're looking for 10x return, 15x return. And so they don't invest. And so what has changed dramatically lately, which has been wonderful for development of antifungals, and which is why we actually have a few antifungals kind of on the horizon, is that the FDA has been advised to exercise maximum possible regulatory discretion. That's the wording in the law. And they've created these, as well as these breakthrough designations that are allowing drugs to come on market faster. So the thing about olorifim is the reason it's going to the market as a phase 2 is they would get approved for a limited indication. It would get approved only for Scedosporium, Scopulariopsis, Lamentospora, these like pan-drug resistant antifungals, fungi, and for refractory aspergillosis, with the idea that they could then be used with the commitment of a full phase 3 and a phase 4 follow-up afterwards. And so that's exciting. And that's actually led to renewed investment because now the amount of money that it takes to bring a drug to market is much less. Plus there's these breakthrough designations that can kind of essentially be sold off. Like there's money that can be made by getting that designation. So the regulatory environment has improved, which is hopeful. But I had a conversation literally last week when I was in a conference Many drugs, because of this financial reason, like for example, Orofim was discovered in 1970s and it got shelved because of one very specific reason. You had no commercial pathway for a drug that didn't both kill Candida and Aspergillus. So anything that killed one, but not the other, doesn't matter how good it was, would just not be developed further. And so in the assays, it doesn't kill yeast. It has zero yeast activity whatsoever, but it was amazing at killing Aspergillus. It just got shelved. It had no commercial pathway. So a lot of it has to do with cash. My favorite analogy to that was written by John Rex. I think you probably know John. Prominent mycologist, been around for decades. He talks about how we need to change our understanding of antimicrobials and what they do. And he uses the sprinkler analogy, the fire sprinkler analogy, which is, did you use the fire sprinkler today? The answer is no. But the answer is yes, because you live in a building, or you exist in a building right now that cannot exist without fire sprinklers. And that's what antimicrobials are. They are the bedrock and the pillar of our modern medicine. What we do cannot be done without antimicrobials, but we insist on paying in them for a per usage structure, which leads to escalating resistance and an never-ending arms race. And so we implement stewardship because our incentives are wrong. And the incentives are, we need to pay for the development as a society of drugs that prop up all of medicine. And then if they are not needed right now, but they have a potential to be needed later, we shelve them. We shelve them now, we don't use them, and then we use them in the future. The problem is if you do that in the current model, you disincentivize development. Yes, exceedingly expensive. Our newest antifungal, Rezafungin, is going to be $4,500 per dose for the loading dose, $2,500 for the repeat dose. Isavuconazole, which is the drug that came right before it, so 2015 was the release date, is still about $9,000 a month. And you may imagine insurance companies are not happy to pay for it if they can try to avoid it, even when it's necessary. Yes. I think some variation of that sentence. So the question here is, do you think increased use of immunosuppression could also lead to rise in fungal infections? Has been used in basically every grant ever written by every mycologist ever, because it just makes such intuitive sense. I'm not sure it's true. And I think the best work that was done to show that is by a guy who's sitting in this room right now. It's actually Dr. Iggy George, who did a large-scale study of Cryptococcus and their changes over time. And what he's shown is that, HIV-related Cryptococcus is going down. Awesome, makes complete sense. But so is non-HIV. And the answer for that is because the patients aren't different, the drugs are different. And so now you're giving rituximab, where previously you would have given like 1.5 to 2 mg per kg of prednisone, because the patient needed something. They would have been given something. And there is more of them who are surviving, but the immunosuppression that we're using is gentler and more tailored to the disease, and it's lower risk for ongoing fungal infections. So it's hard to know because there's that dichotomy. There's more patients because they're not dead, and but there's better immunosuppression that's more tailored. At least for Cryptococcus, that actually means that the number of cases has gone down over 20 years. I think it's worth exploring. The problem is that by the time you get to the point where you have an invasive fungal infection, your immune system is not right. Very few people will get an invasive fungal infection with a normal immune system. Some do. There's plenty of people in my clinic who are immunocompetent, who have crypto or who have histo or blasto or cocci, but not so much aspergillus, not so much invasive Candida. And certainly those are useful adjuncts. And I think that they will improve, that some of them might end up being helpful. We're currently working with developing studies for IL-7 and inhaled GM-CSF for those specific, to test those specific hypotheses. It might work, it might not. Those studies are also really hard to design because the immune deficiencies that lead to fungus, so like the fungus is the phenotype, being infected by the fungus is a phenotype, but the genotypes are varied. And so there might be certain patients for whom adding something like GM-CSF or IL-7, IL-17, anti-PD-1, whatever we want to use, may actually dramatically improve, but there are people who it may dramatically worsen. And so in a clinical trial, it's really hard to demonstrate that unless you know a priori which groups those are and how to segregate them. And so that's the difficulty on that point. There is also a antifungal device that's actually currently in development that basically could filter out the CSF. It's like, those of you who may have had it at the house, it's essentially a reverse osmosis filter that could pass CSF through the membrane, and then you essentially remove all Cryptococcus from the CSF. That could be a useful adjunct along with antifungals because, things like Cryptococcus are not really a meningitis, it's a meningoencephalitis, so you can't really just do that. But it's also a disseminated disease on top of it, right? Because when it's in the brain, it's in a lot of other places. So it could be used as an adjunct, but it's certainly not something that's gonna, I don't foresee those things being the magic bullet. Thank you all.